Turns out, the immune system's secret weapon against cancer. Your cancer cells have been bribing your immune system the whole time, and we finally figured out how they're doing it. The twist? The bribe isn't some foreign molecule. It's not a disguise or a chemical shield. It's a protein your cancer cell makes naturally. One that tells your macrophages, those aggressive immune cleanup crews, to stand down, to stop killing, to let the tumor grow. And if we can block that signal, early evidence suggests we might flip the entire game. So here's the question. What if we've been fighting cancer wrong this whole time, and the answer was literally inside the tumor cells all along? For decades, immunotherapy has worked like a hammer. Whack the brakes off the immune system, release the hounds, hope they eat the cancer before they eat you. And it works, sometimes spectacularly. But here's what's maddening. Sometimes it doesn't work at all. Some cancers laugh at your immune system. They're not hiding, they're not invisible, they're just safe, untouchable. The medical world assumed cancer cells had developed some exotic defense, some elaborate trick we hadn't discovered yet. But what if the truth was weirder? What if cancer had hijacked a system that was already there, one that evolved to keep the peace between your cells? This study, published in July 2026 in the journal Oncogene, decided to ask a different question. Not why does immunotherapy fail, but what specific genes does a cancer cell need to survive when it's under attack from macrophages? And that required a massive methodical hunt through the entire human genome. The research came from a sprawling international team, Zaalberg, Lacoste, Minnee E, and colleagues at the Netherlands Cancer Institute, Fred Hutchinson Cancer Center, and the University of Washington. These aren't small labs. These are heavyweight institutions with genomics infrastructure, robotics screening facilities, and animal pathology teams. This wasn't a curiosity project. This was a war machine designed to find a specific target. And they had a very clever idea for how to find it. So here's what they did. They took prostate cancer cells, human ones grown in dishes, and they ran them through a genome-wide CRISPR screen. If that term feels heavy, here's the analogy. Imagine your genome is a massive building with thousands of light switches. CRISPR is a way to flip each switch off, one at a time, to see which rooms go dark. The researchers systematically disabled genes, one per batch of cancer cells, and then they exposed those cells to macrophages. Real immune cells, hungry ones. Then they watched what happened. Which cells got killed? Which ones survived? The genius part, the cells that survived without a particular gene, that's your hint. That missing gene was probably keeping the cancer safe. Flip it off and the macrophages eat the tumor. Leave it on and the tumor walks free. They ran this across tens of thousands of genes, completely systematic, completely exhausting, completely necessary. Here's what they found. Several genes emerged as critical, but one stood out, the androgen receptor, AR. Now, if you've heard about prostate cancer, you've heard about AR. It's the protein that makes prostate cancer cells respond to testosterone. It's been the main target for prostate cancer drugs for 40 years. But here's what was new. This study showed that AR does something else entirely. It actively suppresses the immune system's ability to kill cancer cells. It's not just fueling cancer growth, it's negotiating a truce with your immune system. When the researchers deleted AR, the cancer cells became vulnerable. Macrophages killed them more efficiently. It wasn't a small effect. It was dramatic enough that it changes how we should think about AR. It's not just an oncogene. It's an immunomodulator, a silencer, a peacekeeper that keeps your own immune cells from doing their job. They validated this in multiple models. Different cancer lines, different experimental setups. They even tested it in mice with actual tumors. The signal held. The effect was real. But here's what matters for you. Prostate cancer treatment has been locked into one lane for decades. Shut down AR, starve the cancer of testosterone signals, watch tumors shrink. And it works. Then it stops working. Resistance builds. The cancer mutates around the drug, and doctors have very few options left. What if, though, instead of just blocking AR's cancer-driving function, you also exploited the fact that AR is making your immune system useless? What if you could hit cancer with two weapons at once? Directly poison the tumor cell with AR inhibitors and simultaneously wake up the immune system to attack. That's not theoretical. That's already being tested. That's the direction this research points. Now let's be honest about what this study can and cannot claim. It's done in cell culture and mice. Neither of those is a human patient. The effect might not translate perfectly. Prostate cancer is complicated. There are many types, many genetic backgrounds. And this study doesn't tell you whether blocking AR's immunomodulatory function would actually help patients in the clinic. That's the next step. That's the real test. But the mechanism is now visible. The target is now clear. And that matters enormously. There's something bigger happening here too. For years, the field has treated cancer cell biology and immunology as separate kingdoms. The tumor-intrinsic stuff, AR, mutations, growth signals. The immune stuff, T cells, macrophages, checkpoint proteins. Totally different teams of researchers, different conferences, different vocabularies. But this study is saying something radical. Those worlds are not separate. Your cancer cells are directly controlling your immune cells through genes we thought were just about cancer. That changes everything. It means that blocking immunotherapy resistance might not require inventing new immune tricks. It might require understanding that cancer has been using the same switches we've been trying to tweak all along. It means the answer was inside the tumor the whole time. We just weren't looking in the right way. This is the kind of work that doesn't make headlines because there's no drug yet, no patient trial, no cure. But it's the work that makes cures possible. It's the work that reframes the problem. And once the problem is reframed, the solutions start to appear. The single most mind-blowing thing about this research is this. Your cancer didn't develop some alien superpower to escape your immune system. It borrowed a tool from your own biology and weaponized it against you. And now we know which tool, which means we can finally take it back. So here's the question haunting the clinic right now. How many other cancers have already solved immunotherapy and we just haven't caught them yet? Turns out scientists have been busy, and they're not done yet.